the scientific name is allium species, and then iodine, and then methylene blue. So the purpose of you using iodine and methylene blue is to dye the cell. Okay, so plant cell and animal cell, within the plant and animal cell, you have the cell will have nucleus. Okay, so for example, okay, so, okay, so this is, for example, a plant cell. Okay, so plant cell will have cell wall and plasma membrane, and then it has a large central vacuole. Okay, and due to the large central vacuole, it has the nucleus, and all of the other organelle will be pushed, okay, to the side of the cell, to the peripheral of the cell. So the nucleus is here, okay, for plant cell. For animal cell, okay, so because there is no there is no central vacuole, so basically all the together in the cell, but the nucleus will be roughly at the center. Okay, will be roughly at the center of. The purpose of you using iodine and methylene blue is to dye the nucleus of the cell. Okay, the nucleus of the cell. So if you dye the nucleus, sorry, if you dye the cell under the microscope, you should be able to observe the presence of the nucleus. Okay, so if you do not use these two dye, okay, you won't be able to observe the presence of a nucleus. You can only observe large organelles, okay, under light microscope, okay, such as nucleus, and also for plant cell, apart from nucleus, you can observe the presence of chloroplast. Central vacuole tak boleh nampak dengan jelas sangat, as it is an organelle that has a fine membrane, and it is to store water, okay, water, to make sure that the cell is in turgid condition. So awak tak boleh lah nampak dengan jelas macam awak nampak nukleus, kan. So tapi awak boleh nampak kesan adanya central vacuole tu dengan the nucleus of the plant cell is pushed to the side. Okay, so tapi kalau under the light microscope, you cannot see other organelles such as ribosome. You cannot see mitochondria. You cannot see Golgi apparatus. You cannot see the presence of mitochondria, vesicle, because all that organelle will be too small for you to observe under the microscope. Okay, so the iodine, the methylene blue will only dye the nucleus of the cell. Okay, so you need a scalpel, you need toothpick, forceps, microscope slide, microscope itself, the cover slip, reticule, and prepared slide of prokaryotes. Reticule ni kita guna, okay, untuk measure cell size. Okay, reticule is a tool used to measure cell size, okay, under microscope. Okay, so these are the procedures. Okay, so we are going to look at the video actually, okay, the demonstration video, which is prepared by Madam Niza. Okay, Madam Niza, for the first experiment. Okay, so please pay your fullest attention to this video. Saya share sekejap. Experiment 2, okay. Okay, this one. Okay, this is the video. Boleh nampak ya?
Boleh.
Boleh. Hi everyone, welcome to Acid Biology YouTube channel. Today we are going to watch a demo video on experiment 2, cell staining and observation. In this experiment, we will observe 3 different types of cells, which include plant cells, animal cells, and bacteria. We will first run through the materials and apparatus that we are going to need. Next, we proceed with preparation of slides and staining process. Finally, we are going to observe the slides under the microscope using the proper technique. But before we continue, don't forget to like this video and subscribe to this channel. Thank you. So, let's continue with our experiment. So first of all, let's start with the materials and apparatus needed. There are 3 materials needed in total. They are onion, iodine, methylene blue. 6 apparatus to complete this experiment. The first one is scalpel, forceps, cover slip, microscope slides, toothpick, and a microscope. Now, let's start preparing the slides. For plant cells, we are going to use onion epidermis and iodine as staining.
This is James. As a company HR, he has to process all these employee benefits claims manually, and it's really stressing him out. What he didn't know is that there's a better life.
Cut a small section of an onion using a scalpel. Use forceps to remove the cutting. And carefully peel off the thin, transparent epidermis. Transfer the epidermis in the center of microscope slide. And then put a drop of iodine stain on the epidermis. This is a wet mount. Place one edge of cover slip to one side of stain and slowly lower the cover slip. If you need to remove excess stain, touch the edge of slip with paper towel. For animal cells, we are going to use cheek cells and stain them with methylene blue. Use a toothpick to gently scrape inside of your cheek. Then smear and mix the cell in a distilled water. Next, put a drop of methylene blue to stain the cell. Place one edge of cover slip to one side of the stain and slowly lower the cover slip. Now, let's continue with slide observation. You will learn the right way to use a microscope, so make sure to set 100% attention to the demonstration. We will use the plant cell as example. Microscopes usually are stored in a dry cabinet. If you need to get the microscope, be sure to hold it right. Unwind the electrical cable and turn on the electrical power of the microscope. Unscrew the ocular lens and remember to adjust it to the placement of your eyes. Switch on the microscope and remember, do not use the fine focus knob. Use coarse knob to adjust the stage so that the stage is at the highest position. Place the specimen slide on the stage and secure with slide holder. We will start off with alien slide. You can see the stage knob can be adjusted to move the slide front, back, or left and right. Look through the eyepiece and make sure not to use fine focus knob at this moment. Move the coarse knob until the image comes into focus. And then you can use the fine focus knob to make sure that the image is clearer. This is what you will see under the microscope. change to the next objective lens. Once 10 times objective lens is clicked, adjust the sharpness and clarity using only fine focus knob. Please remember that at this point, you cannot use coarse knob. You need to adjust the condenser and light intensity until the image is clear and bright. This is what you will see under the microscope at 100 times magnification. You can use the stage knob to move the microscope stage so that the specimen is in the center of the field of view. Do the same with the 40 times magnification power of your objective lens. Please remember to only adjust fine focus knob. Remember, you cannot adjust coarse knob at this point. Again, you can adjust the light intensity so that the image is clearer and brighter. This is what you will see under the microscope at 400 times magnification. You can adjust the stage knob so that the image is at the center of your field view. If 1000 times magnification is required, this is what you need to do. After getting a clear image at 400 times magnification, turn the barrel of the microscope so that the 40 times and 100 times lens are halfway between and directly over the slide or cover slip. Next, you need to place a drop of oil in the middle of the slide or on the cover slip. Carefully turn the objective lens until the 100 times magnification lens contacts the oil and snaps into position. You can adjust sharpness and clarity using fine focus knob. After the observation, remember to wipe off the immersion oil and clean the objective lens with lens tissue. We already observed our plant cells under the microscope before. Now, let's see how animal cells look like under the microscope. This is chick cell under the microscope at 40 times magnification. This is chick cell under the microscope at 100 times magnification. This is a chick cell under the microscope at 400 times magnification. This is how bacteria looks like under the microscope at 400 times magnification. The bacteria is circular shape and it is stained in purple. Well, students, that's all for experiment 2. Do complete your observation and don't forget to submit your lab report on time.
Okay, so that is basically the demonstration for experiment 2. So for your lab report, okay, you have to prepare a lab report where you wrote the title, the objective, the introduction first, the objective, and then the materials and the method, and then for the results. Okay, for I want to, sebab yang awal tu senang je. For the result of the experiment, you have to include the drawing, okay, of the cell observed under different magnification. Okay, so you have to draw uh, the 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 structure of the plant cell, the structure of animal cell, and also the bacterial cell at different magnification. Saya tak nak you all include a screenshot of the picture. Itu sesiapa pun boleh buat. In science kan, you need to have the ability to be able to observe and draw. Okay, observe and draw. So okay, so for your observation, for example, when you are observing
Hi
for your
everyone,
result,
welcome to S.
okay, so result tu saya nak awak ambil image lah kan. Observe this one, observe under what magnification. So you draw your drawing, okay, so your drawing should be each drawing for each magnification should be not smaller than half a page. Okay, not smaller than half a page. So maksudnya kalau ini apa? Magnification bawah
Focus knob to make sure
ini bawah 40 ni kan. So kita ada 40, 100,
change to the next objective lens. Once 10 times objective lens is clicked, adjust the sharpness and clarity using only fine focus knob. Please remember that at this point, you cannot use coarse knob.
okay, so magnification tu kita ada 40, 4 kali 4, 10, 40 kan. 4, 10, 40. So for each of this magnification, you have to draw your drawing. So Madam Liza dah bagi dah image for each stage tu. So you just pause kan, and then you draw. Okay. So when you are drawing your image, so make sure you draw it properly lah kan. So as you can see from this image, so you take the whole scene here kan. So you draw the plant cell. Okay, you draw the plant cell with the nucleus at the center. So as you can see, this plant cell is dyed using iodine. Okay, so the iodine will dye the nucleus a lot clearer in this brown, yellowish brown color. Okay, so you should be able to observe the presence of the nucleus. Kan, so kat sini nampak the nucleus here kan. So as you can see, most of the nucleus are located at the peripheral of the cell. Ini nampak macam tengah tapi sebenarnya it is pushed okay to the side. Okay, sebab ini kena imagine lah dia dah bentuk sel ni dia ada volume lah kan, dia ada volume. So the central vacuole akan push to the side tapi nampak dia macam at the center here, tapi actually it is at the side of the cell. Nampak kebaikan dia at the side due to the presence of the large central vacuole. Okay, so make sure when you draw this cell, you draw the cell with a thick layer here. So this will represent the presence of cell wall. Okay, so you label inside, you have the plasma membrane, and then you have the cell wall, and then inside you have the cytoplasm, the nucleus, and then maybe you can also highlight the presence of central vacuole. Okay, central vacuole. So those are the structure that you need to include in your drawing and also for your labeling. Labeling your diagram will have marks kat situ kan. So a neat diagram, okay, a neat diagram of your observation. Labeling the diagram will gain you marks. Kalau awak draw tapi tak ada label, tak ada markah. Okay, so make sure you draw each of this drawing at least the size is half a page. Okay, half a page. And then the drawing must be done in pencil. Okay, but the labeling you can label in pen. Okay, so you don't have to, you are not required to shade your cell. Just draw a sketching of your observation. Okay, a sketching of your observation using pencil. I don't want you to color the cell. Okay, just sketch using pencil. So you also have to state in your drawing the magnification used to observe the specimen. Okay, so again I repeat, for your result, okay, your drawing must be not smaller than half a page. You must include a title for your drawing. Okay, so for example, plant cell allium species. Okay, and then your drawing, your labeling, and then at the bottom you state the magnification used to observe the image. So I want you to draw your observation for each of that magnification. 4, 10, 40 times magnification. Tiga. Okay, tiga stage tu. Alright, and then same goes to the animal cell or chick cell. Okay, kalau kat sini awak nampak dia Madam Liza, kalau kat mikroskop tu dia akan label dia ada 4 lens. Kan 4 lens, 4 kali 10 kali, 40 kali, 100 kali. Tapi kali tu kena kalikan dengan 10. So dia akan jadi 40, 100, 400, 1000. Macam tu kan. So magnification tu awak state lah mana satu. Is it 40? Is it 100 times? Is it 400 times? Or is it 1000 times magnification? So make sure you write the magnification correctly. Okay. So okay, so this is the image observed under 40 times magnification. So you draw. So as you can see here, these are the. Oh, nampak saya point ni? Nampak ke tak saya punya arrow dia? Nampak?
Nampak.
Okay. So as you can see, so this is the chick cell. So this is the individual chick cell. Satu, dua. How do you know that it is the chick cell? Because you can see the nucleus at the center. So for this chick cell, it is dye using methylene blue. Okay, so kalau tak dye using methylene blue, you cannot observe the cell clearly. Okay. So this is one cell, this is another cell. Yang lain-lain ni, cebisan-cebisan ni tak payahlah kan. So you just draw the clearer ones. 1, 2, 3 ni. Kan ini dia tak nampak jelas sangat. Maybe the cell has been ruptured during the process. Kan ini pun macam rupture. Yang ni you should see the cell clearly lah. 1, 2, and 3. Okay. So this one is under 400 kalau tak silap. Ini awak tengoklah balik Madam Liza punya explanation kan, state the magnification. So this one you can see the image for the cell much clearer. Okay, so you have one whole cell, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So kat sini okay, kita dah belajar for chapter 2, towards the end of the chapter, kita dah belajar pasal animal tissue, right? Okay. Animal tissue, we can classify animal tissue based on the shape of the cell. So for this animal cell, what is the shape of this animal cell? A plant, chick cell lah in this case. Kita tergolong bawah animal lah. Kita ada plant and animal. Kan? Tak kalah plant. So animal lah. So the type of cell. What is the type of cell of this chick cell ni?
Epithelial cell.
Okay, jenis dia epithelial cell. What do you understand by epithelial cell? Cells that forms the lining of organs kan? The lining of organs, the cavity inside your body. It forms the lining of cavities and also the lining of organs, and also the outer layer of your skin. Is it?
Yes.
Okay. So epithelial adalah jenis tisu. Okay. Tapi dalam epithelial cells tu, epithelial tissue tu ada 3 jenis sel. Apa dia? Kita ada 3 jenis. Squamous, cuboidal dengan
Stratified.
okay, certified lain. Columnar. Okay, apa beza dia? Please sambil ni awak tengok apa yang nota. Shape of the epithelial cell so ada 3, columnar, squamous dengan cuboidal. Okay. So in this case of the chick cell, dia jenis yang mana?
Squamous.
Squamous. Kan, squamous kenapa? Squamous, awak ambil daripada perkataan asal dia, squished, flat. Nampak tak? Sel dia nipis-nipis. Sebab tu dia boleh form linings. Faham?
Faham.
Okay, so dia adalah sejenis
Faham.
squamous. Lepas tu kita tengok cara susunan sel tu. Cara susunan sel tu ada 3 cara. Satu apa? Simple, stratified, pseudo
Stratified.
pseudo stratified kan. So simple tu apa maksud dia? Single layer. Okay, single layer. Single layer of epithelial cell. Okay, yang itu awak boleh jumpa dia apa? Lining, it forms the wall of alveolus kan. Dia kena single layer of flattened sac sebab apa? Dia nak allow diffusion of respiratory gas. Okay, sebab tu simple squamous. Okay, so kita ada contoh lain, kita ada stratified. Kan, stratified maksudnya it forms layers of cell. Okay, layers of this flattened sac. So yeah, in this case of in the oral cavity kan, the tissue that lines the oral cavity consists of squamous epithelial. Squamous, stratified squamous epithelial cell. Betul ke tak? Lining your cavity. Yang mana satu? Pseudo-stratified, simple or stratified? Single layer, many layer or many one layer that seems like many layers? Boleh recall balik tak ni?
One layer.
Is it one layer? Lining your oral cavity. Kena single layer je
It's
ke?
pseudo.
Okay. Can you all refer to your notes, slide 90, please? Dah buka?
Dah.
Dah. Okay, awak tengok kat situ. Stratified squamous epithelium. Okay, example, the outer skin, the lining of mouth, okay, anus and also vagina. So in this case of oral cavity should be stratified squamous. So sel-sel dalam awak punya inner lining of mouth tu kena banyak layer. Kan, kena banyak layer. Sebab apa? Your mouth is exposed to the saliva containing amylase that has lysozyme. The condition will cause the cell at the surface will always
Pseudo-stratified.
will always be replaced. Bukan pseudo stratified, stratified. Many layers. It forms many layers of flattened cells. Faham tak? Ni awak nak exam ni minggu depan pasal chapter 2. Okay. So slide 90 of your notes. Okay, awak tengok It forms many layers kan. So the inner lining, the lining of your mouth is stratified squamous. Sebab apa? It has to form many layers of the epithelial cells so that the exposed layer kan, the cell at the surface can always be replaced with new one. Sebab apa? Like the oral cavity will always be exposed to your enzyme, amylase, the saliva kan that has lysozyme. So dia akan selalu sentiasa terhakis. So mesti kena ada replace dengan sel-sel yang baru daripada lapisan yang bawah. Sebab tu kena terdiri daripada stratified squamous. Faham Kalau pseudo stratified, apa yang dimaksudkan dengan pseudo stratified? Pseudo stratified tu it is a single layer but it seems like many layers because the cell is composed of cell with with varying heights, with varying cell size. Dia nampak macam banyak layer tapi dia satu layer sahaja. Macam contohnya, kalau ada gambar kat sini, ada tak? Kalau sudah stratified. Nampak? Okay. So consists of a single layer of cell varying in height. Dia nampak macam banyak layer tapi dia sebenarnya single layer sahaja. Okay, single layer saja sebab sel-sel dia terdiri daripada sel-sel yang panjang, yang pendek, macam tu, yang kecil, yang besar. Tapi sebenarnya dia adalah one single layer. That is pseudostratified. Okay, so in this case of oral cavity, it is stratified squamous. Okay, so kena faham lah mana nak apply istilah-istilah ni. Okay, so please tengok balik awak punya nota, buat revision balik lepas ni. Alright, so we are going to proceed with Okay, so saya share balik semula. Okay, so this is for the animal cell. So you draw the image for 40 times magnification, 100 times magnification and 400 times magnification. Okay, labelkan kesemuanya. Okay, so this one should be 40, this one should be 100. Okay, so this one should be 400. I'll check balik. Okay, dengan dia punya explanation. So this is bacterial cell. Okay, so this is bacterial cell. I'm not sure what is the source of this bacterial cell. Selalunya kalau untuk dalam eksperimen, kita akan guna daripada yogurt lah. Tapi ini source dia saya tak sure. Tapi Madam Liza tadi kata bentuk dia spherical. Okay, so kalau bacteria, the bacteria that is spherical in shape, kita group dia sebagai cocci. Nama group bacteria tu as cocci. Okay. Tapi kalau dia panjang-panjang, kita namakan dia sebagai bacillus. Kan, road shape. Okay, so tengoklah kat sini just spherical shape macam tu. Okay, bulat-bulat. Okay, so that is for experiment 2. So for your result, you draw your drawing for each magnification and then you label neatly and then you discuss. For discussion, discuss lah. You discuss the observation, kan? So you discuss the shape of the plant cell, the shape of the animal cell, the component of every cells. So you discuss in your discussion and then you do your conclusion and then list down a few references. Okay, so kalau awak tengok dekat dalam awak punya lab manual ni, kan, dia ada bagi kat bahagian awal-awal lab manual ni. Okay, so these are the components of the lab report that you must include. So saya tak tengok awak punya lab report untuk mini project macam mana. So it should consist of this component. Okay, you must have your title, your abstract, your introduction, materials and method, result, discussion, conclusion, references, appendix, optional. Okay, tak ada pun tak apa. So conclusion, how do you do a conclusion? You answer back, okay, the objective of the experiment that you wrote in the introduction part. Okay, so introduction, you have your introduction. At the end of the introduction, you write your objective. Okay, so conclusion, awak answer balik, you answer back the initial objective of the experiment. So did you manage to achieve the objective of the experiment through the data that you get from the result? Kan? So apa objektif dia, awak tengok balik, awak jawab balik objektif tu. So reference, kan? So reference ini adalah cara you write reference. Okay, so kat belakang ni ada contoh, kan? So awak boleh google lah macam mana nak tulis reference. Tapi sebenarnya how to write reference is that, for example, if you are referring to a book, okay, so this is the authors, okay, so you have to write down the authors of that book, okay, the title, so sorry, the year the book is published, the title of the book, okay, the page that you refer to, and the and the publisher. Okay, and the publisher of the book and the country the book is published. Okay, itu kalau refer kepada buku lah. Tapi kalau refer kepada artikel dalam internet, so macam ni lah awak buat. Kan? So siapa yang wrote artikel tu, tahun bila, apa title artikel tu. Ini kalau awak refer kepada website yang dah established, contohnya NCBI, website yang established daripada certain university punya website. So you write down the website address and then you write down the date that you access the reading material, the article. Okay. Tapi kalau awak access kepada journal, artikel dalam journal, sama juga, you write the authors, the year the article is published, the title of the article. Tapi kalau di dalam journal, okay, you don't have to write the website here, but you write the journal, the journal punya title. Okay, kat situ. Sebelum journal punya title kena ada page lah kat sini. Okay, kena ada page. Saya tak sure, Madam Huda ada terangkan tak macam mana nak tulis reference?
Ada.
Okay. So lebih kurang macam saya explain ke? Okay, so this is an example of how you write a proper reference. So untuk eksperimen Sal ni, please include at least 2 or 3 references, okay, that you write properly. So references will also contribute to marks lah, kan, proper referencing. Okay, so that is for experiment 2. So far, do you have any question for the lab report for experiment 2?
No.
Okay, so we can proceed with experiment 3. Sekejap. Okay, so this is experiment 3 I'm going to share. Buka ini, siap. Okay, this one.
Experiment 3. Investigating cell membrane permeability using turmeric, Curcuma longa. This is the last experiment for this semester. In this experiment, we are going to investigate the effect of ethanol on the surface of cell membrane of Curcuma longa. Now, let's take a look at the materials needed in this experiment. We need turmeric or Curcuma longa, ethanol, and distilled water. Now, let's take a look at the apparatus needed. We need cork borer, scalpel, ruler, forceps, beaker, and measuring cylinder, not to forget a white tile. Use a cork borer to cut 6 cylinders of fresh Curcuma longa tissue. Hold the turmeric with your left hand tightly, and by applying force, I mean a lot of it, start the process by pushing down the cork borer through the tuber. Take out the borer and then use the solid pin to remove the fresh cylinder tissue out of the borer. Use a ruler and a scalpel carefully to cut the cylinders so that they are at the same length. You can clearly see in this video we use 2 cm measurement for each cylinder. Cut it gently and place all cylinders in a beaker of water. Let the cylinder soak in water for 5 minutes. Make a 25% dilution of ethanol. Total volume needed is 10 ml. So, in this video, we use 7.5 ml of distilled water and 2.5 ml of ethanol. Label 3 small beakers respectively. One with 100% ethanol, second one with 25% ethanol. As you can see, we are mixing the dilution now. And finally, a beaker labeled distilled water. Fill each beaker with its respective solution of 10 ml each. Remove all cylinders from water and place onto a paper towel to dry. And then place 2 cylinders in each beaker. Let stand for 15 minutes. After that, we observe the color change of the solution in each beaker. This is the end of this experiment. Now, you need to record all your observation in a datasheet experiment. A datasheet must consist of a table to record color change of 3 different solutions used. Remember, this is the initial color of 100% ethanol, 25% ethanol, and distilled water. They are all colorless. After 15 minutes, the solution changes in color. This is 100% ethanol, this is 25% ethanol, and this is distilled water. Describe the color. You may also put this figure in your datasheet experiment. Finally, you need to discuss all of the findings. Why do you think the color changed? What happened to plasma membrane in reaction to alcohol? After completing your lab report, don't forget to submit it in your Google Classroom on time. Good luck!
Okay, so that's the demonstration video of experiment 3. So basically, if we look at for the experiment 3, sekejap, how do you go back? Okay.
Experiment 3.
Okay, so experiment 3, investigating cell membrane permeability using turmeric. So if you look at the turmeric, it is yellow in color due to the presence of pigments. Okay, so in your discussion, okay, you must identify, I want you to find out the name of the pigment, of the yellow pigment that are found in the turmeric. Okay, so um, so the procedure involve you to immerse the turmeric cylinder in different solutions. So you have water, you have diluted alcohol and concentrated alcohol. Okay. So from the result obtained, you observe that okay. So for this water, the color of the solution is really, kalau tengok kat sini kan, it is clear. Okay, so it is clear. As compared to the solution of dilute alcohol 25%, the color is light yellow. Okay, compared to 100% of ethanol, it is bright yellow. Okay, bright yellow. So why do you think that the color is different in this different solution? Kenapa air jernih, 25% etanol, warna kuning cair. Tapi kalau 100% warna kuning pekat. What is the effects of ethanol towards the membrane of the cell for that turmeric? Apa effect? What is the effects of immersing cells in ethanol?
The water from the cell will move out.
Okay, water. Okay, sekarang ni kita nak tengok warna solution tu. Warna solution tu ada jadi jernih, ada jadi kuning cair, ada jadi kuning pekat. Dia kat sini, if you relate your discussion with the process of water moving in and out of the cell, which is osmosis, is not correct. Dia bukan melibatkan you immerse in a solution of that contains salt or ions that cause the solution to be what do you call it as saline solution kan. Kalau yang menghasilkan solution hypertonic, hypotonic, isotonic tu, this is not the case kan. You using water, diluted ethanol, concentrated ethanol, it does not apply to the concept of osmotic solution hypo salah, sebab kita bukan menggunakan gula ataupun garam ataupun other salt as solute. Tapi kita melibatkan konsep apa kat sini? Dia bukan melibatkan pergerakan air.
The movement of solids in the cells.
The movement of what?
Solutes.
Solute. What solute is present in this solution?
The yellow pigment.
The yellow pigment comes from the cell. Kunyit ni, turmeric ni, dia warna kuning disebabkan oleh adanya pigment-pigment warna kuning dalam sel ni. Bila awak immersekan dia dalam solution yang berbeza, dia akan menyebabkan pigment dari dalam sel keluar daripada sel ke dalam solution. So that's why you will observe different intensity of the yellow color. Okay. So sekarang ni apa effect macam Madam Liza tanya tadi, what is the effect of the ethanol towards the membrane of the cell? What is the component of plasma membrane? Plasma membranes.
Phospholipid bilayer.
Phospholipid bilayer. Phospholipid bilayer is grouped under lipid, right?
Yes.
What is the solvent for lipid?
Ethanol.
Kan? Ya ke tidak?
Yeah.
Kita tengok apa yang nota.
Madam,
Yeah.
is that the plasma membrane will dissolve in ethanol? That's why the pigment can flow out.
Yes. Okay. So the effects of immersing turmeric will cause the membrane of the cell to break. Okay. To break apart. Okay. The ethanol will cause the membrane of the cell to break apart, and the organelle, okay, that stores the pigment, the membrane of the organelle that stores the pigment to also break apart. Nanti bila awak buat reading, awak buat pembacaan, satu awak cari nama pigment, dua awak cari kat mana pigment ni store. Okay. So the pigment should be stored in an organelle that has membrane. So the effects of the ethanol is to, one, break the membrane of the plasma membrane, two is to break the membrane of the organelle that stores the pigment. So once this membrane has been broken down, okay, by the ethanol, it releases the pigment into the solution. So that's why when you observe the image for the observation here, you see that for water, the color does not change. Okay, so because water is not the solvent for lipid, so the membrane of the plasma membrane will remain intact. Okay, so the pigment will cannot diffuse out into the water. But if you immerse in a very dilute, which is 25% of diluted ethanol, okay, so the ethanol, the dilute ethanol will break, okay, will break some of the cell. So that's why you see some of the pigment diffuse out and the color will turn into a light yellow color. Okay, the intensity of the color is light yellow. Okay, but if you immerse in a very concentrated ethanol, which is 100%, okay, so it gives a very bright yellow color. So it shows that many of the membrane, okay, has been broken down by the ethanol and releases the pigment into the solution and causes the solution to turn into intense yellow color. Okay, bright yellow color. So remember, this concept does not is not related to isotonic solution, hypotonic solution, hyper iso hypo. Okay, so sebab dia bukan melibatkan larutan garam. Okay, right. So itu awak kena faham konsep. Kenapa kita immerse dalam ethanol? Because ethanol serve as the solvent for the lipid. In this case is the lipid, phospholipid bilayer. Okay, so faham
Faham.
Okay, so for your result, you have to construct your own table to record the data, of which in the table you must state, okay, one column you have the different solution, kan, 100% ethanol, the 25% ethanol, the distilled water, and then you have a column to record the initial color, okay, and a column to record the final color for each of the solution. So construct your own table to record the data. You can also include in the result the image. Okay, this image as for reference lah, to refer. Okay, for reference. So in the discussion, macam saya cerita tadi lah, so discussion, you relate your result with the theory. Okay, with the theory like what I've explained. With further reading lah, with further reading that you will refer later. And then after you have done your discussion, conclude the whole experiment, refer back to the initial objective, and then finally is the reference. Okay, so that is the report for experiment 3. So do not combine lab report for experiment 2 together with experiment 3. Do it separately. Okay, in a group. Saya nak awak buat report ni sebagai satu kumpulan lah, bukan individual report. Okay, and then after you have done your report, you submit the report in the drive that I will share with you later. Faham?
Faham.
Okay.
Madam,
Yeah.
for experiment 3, result ni kita orang kena lukis ke kita orang boleh ambil je gambar ni?
Ini boleh ambil sebab ini bukan require for you to observe structure. Faham?
Faham?
Okay, so yang sel tadi je awak draw. Yang ni awak boleh ambil screenshot, tapi you have to record the data in the form of table as well.
For the method, prosedur semua kena lukis ke?
Tak. Procedures for experiment 2 and experiment 3, you write macam kat dalam report tapi in past tense, because it is a report. Report ni adalah laporan benda yang telah dibuat, kan? So you write the procedures in past tense.
Okay, madam. Bila lah submit this experiment?
Week 8.
Okay.
Week 8 on Wednesday. Seminggu lah kalau kira tolak minggu exam, minggu cuti, sebenarnya you are given 1 week, kan? So kita ambil minggu ni minggu 7, you submit at week 8 pada hari Rabu. Okay?
Madam, for both experiment ke experiment 2 dengan 3? ke macam mana yang kena submit week 8 tu?
Both
Okay.
experiment. Okay.
Thank you.
Alright. So any other questions? Okay, so video ni awak boleh refer balik dekat dalam channel lah, Acid Biology channel. Boleh cari kan? Boleh cari tak?
Boleh.
Yeah, so boleh cari balik dekat Acid Biology channel for you to get the data lah. Alright. So if you don't have any questions, so this is that's the end of our session. Okay, so thank you for your attention. So you can leave this session. Kita habis awal sikit lah sebab itu je lah kot untuk kita punya session, unless if you have questions. Okay, kalau tak ada question, boleh you can leave the session. Okay, so thank you class.
Thank you, madam.
Okay, alright.
Madam,
Yeah.
nanti recorded lecture ni nanti madam share dekat drive ke?
Tak. Boleh lah nanti saya bagi.
Okay, thank you madam.
Alright.